is this initiative, the SciComm Synapse?
So SciComm Synapse, like we started recently around 1 year ago in huge gap between the research and also the students. And also in universities, when you go, like people actually don't really understand. They want to pursue research, but they don't understand how to do it. Exactly. So we thought that okay, we can start like a small podcast with scientists, and we can actually invite them and ask them that how did they build their journey, and motivate the other students at the same time. And we have been quite open with inviting speakers, and like we like our average registration for the participants is like 100. So now we are planning to also register this community and go ahead for funding. So maybe from India Bioscience or Science Psychom India or something like that, so that we can do much more better.
Yeah, it's a good initiative,
Yeah,
especially
and
mm,
yeah, please go ahead.
especially with the university system in India. It is really uh, different. On average, we can say it is difficult. Depends upon university to university, but on average, it is very difficult on the university
No,
system to work.
yeah, that's true.
Mm.
So that's why we started this online, I would say online one-to-one podcasts or something like that, because not everybody has accessibility to reach to one place.
Yeah, that's true.
So so that's why, yeah. And Satya told me about you,
Mhm.
and we were really looking forward to have a talk, because we were making a list of speakers
Mm.
among our members. So he talked about you, and then we were like, okay, we are like trying to fix all the speakers. Recently, we also had Rohan Khadilkar from Extract, and also Felix Bast from from Punjab University. So those events were really good. I hope today's event would also be really motivating, especially for the women in science today.
Yeah, I actually didn't prepare. I thought it's just a casual discussion and
Yeah,
fun,
yeah,
which I do with my students. So there was
yeah.
nothing to get ready or to prepare. Satyat told me some questions. I said these are general. These things on the regular basis I discuss. So I don't
Yeah,
think there's something I should prepare, or
yeah,
I would
yeah,
be quite natural way of speaking what I am on the regular basis.
yeah. Absolutely, absolutely. So, do you also have a short presentation or something like that?
I actually didn't prepare a short. I have clubbed the slides. I can skip and show what is relevant because
Okay,
in research, this is a lot of two. But just to give an overview, I have my slides. I can skip a few and show a few like
sure.
this. Not specifically prepared for this particular.
Yeah, yeah, yeah,
But
yeah,
I
yeah,
have
yeah.
clubbed my two presentations into one kind, so to give a
Mm-hmm,
glimpse of something which may motivate people. So
mm-hmm,
I'll be skipping and then jumping to the required slides.
mm-hmm. Yeah, yeah, yeah. Absolutely, absolutely. Then, maybe we can give you like 10-15 minutes
Yeah,
for that. Yeah.
yeah, yeah.
Okay. Okay. That works. So,
Mm.
Somilia Mishra, Bhakti, and like our team consists of like 5 to 6 active members,
Okay.
who are responsible for like different things, who are setting up. And Somilia is also doing his PhD in Baba Atomic Research Center, Mumbai,
Uh-huh.
and Bhakti is a undergraduate student in MS University, Baroda.
Okay. So all age groups. That's what a
Yes,
team.
yes, that's true. Okay, so Milia, we can go live. Let me know.
Yeah, we are live already.
Ah, that's great. Fantastic. Okay. So let's start the today's session with Dr. Vandana.
Okay.
So I welcome you, Dr. Vandana, today for the Psychom Synapse talk for the interactive session from the Psychom Synapse. And me, Dhairya Rajguru, will be the today's host. And we have our team and also the participants. So I think without any delay, we should start the talk because people are really waiting to hear you. And so I request Bhakti to please introduce you to our audience today. So Bhakti, please, can you give the kind introduction for Dr. Vandana?
Yeah, sure. Thank you. So, good morning, everyone. Let me quickly introduce our today's speaker, Dr. Vandana Vinayak, ma'am. She is an esteemed scholar and seasoned professional in her field. She has done her master's in biochemistry from Punjab Agricultural University, Ludhiana. She did her PhD in biochemistry from Kurukshetra University, Haryana, while she was employed as project fellow in a national oilseeds and vegetable oils development board project on micropropagation of potential tree-borne oil plants, protocol development for multiplication in Jatropha, under the supervision of plant biotechnologist Professor A.K. Dhawan. During her PhD, she worked on callus and suspension cultures from 31 interspecific hybrids of saccharum to check their high sucrose content via refractive index, polarimetry, radioactivity, enzymes, protein, and PCR markers. Thereafter, she joined State Forensic Science Laboratory, Haryana, as Senior Scientific Officer Biology and gained knowledge on use of plant algae diatom and its importance in death due to drowning cases. During this period, she solved about 4,000 criminal cases and about 5 cases of national importance. She had been awarded with Young Scientist Award, Meritorious Laureate Award from various scientific organizations, and Noel Dear Gold Medal Award from His Excellency President of India, Dr. APJ Abdul Kalam. In 2023, she has been listed as top 2% scientist category by Stanford University, published by Elsevier publisher Metrics. Currently, Dr. Vandana Vinayak works as assistant professor at Department of Criminology and Forensic Science at Dr. Hari Singh Gaur Vishwavidyalaya, Madhya Pradesh. Her research is focused much on diatom, nanoengineering, and metabolism. She looks onto different aspects to harvest biofuel from diatoms and use of diatoms as nanomaterials in the different fields of bio-nanotechnology. So this was all. Thank you.
Thank you, Bhakti, for the very kind introduction of Dr. Vandana Vinayak, ma'am, today. So, without wasting much time, let's move ahead with the session today. And here I would like to ask like the first question, since Bhakti mentioned about you being your upbringing was in the army family as well, and how did it impacted your academic journey and also maybe the research interest did like since you also were engaged with forensics and criminology, that's why.
As such, like being in the army background and science is not any relation. But yes, since childhood we had a very strict discipline at home. Right now also, so the focus was there on certain things that we have to do for, let it be studies. So we have to do uh, that too very seriously. So I was good in studies right from the beginning. So I mean, always on the merit. So that was not an issue to pursue science as one of my favorite subjects. So it was a passion to do science right from the beginning. And while I was doing PhD, So there was some openings in the forensic science laboratory, Haryana. So I applied there, and that was on the contract basis. So I got the position. It was on the contract, and as I told you, the motivation and the discipline has always taught us to obey the seniors and do your work very dedicatedly. So we had a lot of pendency during those times in the laboratory. Like the cases 20 years back, they were pending in the laboratory. So there was a drive. I remember Director General of Police V.N. Rai sir was there at those times, and he said that we have to, bring the pendency to zero, and for that we have to work 8 to 8, 8 in the morning till 8 in the night. So there was a drive, there was a passion. Everyone in the lab was at 7 in the lab and going 10 or 11 by night. And that drive, again, it filtered few who were so very dedicated. So this was 2 or 3 people who were, including me. So we got, some scholarship from kind of 10,000 rupees and a meritorious award from Director General of Police for working very dedicatedly. So that passion of doing science was always there. And during those times, seeing my sincerity and hard work, then there were regular openings, regular job openings in the lab. So there were two kind of job openings. I had almost completed my PhD during that time, and one was of the lower grade, that was a scientific assistant, and another was of the senior scientific officer. Here I would like to highlight very important lesson for the kids, the growing generation. You should know your value. That is what I want to tell over here. Now I tell you what happened. So there were two job openings, and literally after doing PhD, you need a job, obviously. So I just applied, thinking that I might go for higher studies, more research. But since the openings were in the same lab, so I applied for both the scientific assistant, that was a lower grade, and for the senior scientific officer, that was a commission job. So there was a lot of pressure, as you always know in the government job. There is always a pressure of things, so many which I can't say. So I was sure that I will not get selected in any of them, though the interviews went well. So I was sure I will not get into any of them. And while being in the contract job only, I started working my research in the lab. And the director of the lab that time was Dr. M.K. Goel, and he motivated me. See how motivation is very much essential for anyone to take you higher. Without, like, that's a very, even if the student is good in studies, you don't motivate, he will lose his interest. If it should be there either from the parents or from the teachers. So Dr. M.K. Goel, he told me that since he joined the lab, he wanted to work on this organism known as diatoms, but due to so many other things, and he couldn't for some reason. So he wanted me to do, seeing the sincerity and the hard work, all those things. Again, the senior says something to you, you have to obey. In Forge, that is a rule. So I took that as a thing that since senior has put so much trust on me, so I should do. So I explored this, and I banked one research project from DST, and that was a small project around 5 lakhs, and I got it. So now simultaneously, when the regular jobs were, I had applied for the scientific assistant and the senior scientific officer. That was the second year of my project also. So now the director of the lab said, if you didn't get select in this two jobs, you will have to leave. I said, yes sir, I have to leave. That's no option. And he was so much kind of interested in that project which I had brought, and the project will also go. I said, sir, the project might stay here, somebody else might take over if he or she is expertise in that. He said, nobody is expertise, only you who are expertise, you are taking so much pains. So anyhow, he said, okay, if you get essay position, will you join? He called me in his chamber and he said, if you get, somehow he must be thinking that essay to mil hi jayega. She is PhD, so scientific assistant to mil hi jayega isko, and government job, so she might say yes. So now the point was, I should say yes or no. So I said clearly and bluntly, with a pinch of sorry, I said, sorry, sir, I will not join SA. If I'll join the senior scientific officer post only, which, at least matches my level of education. So he said he was very frustrated because he knew that this is not going to happen. So I went back to my place. I belong to Punjab, Jalandhar. So I went back to place and joined some, biochemistry lab, pathology lab doing. I knew that this is not going to work in government sector because I don't have any, things at higher level. So, but uh, one fine day, I got a call from the director. He said, Varna, you are selected. And I was just driving the car and I was okay, normal, happy. I just put a brake in the car. I was happy. But more than me, he was happy. So I realized when someone motivates you, the teacher, the guide, he is really happy when you progress. So these are the things which I learned from my seniors, not only he, my supervisor, as she read, Bhakti read, Professor A.K. Dhawan. The student teacher relation, people say, remains till the 4 years or 5 years of the PhD. But I'm so blessed that till this age, till this gap of time, my supervisor and my relation has not changed a bit. He is there for me till now. So that's the point of motivation.
I think that is something every PhD student would be looking forward to keep a relation, or anybody, let's say in university as well, your studies, or the director of your first job.
There's a legacy you have to, give it back to your student. What you have got, obviously, it is both ways. What you give, and the student also gets motivated and does the work. It is not like
Yeah,
that way giving. It is both ways.
yeah, yeah. That's true. That's certainly true. So, since you mentioned that you also did your PhD from biochemistry background, so what motivated you firstly to pursue biochemistry? Because I think during early 2000s, or even before that, biochemistry. I mean, yeah, there were people who were doing PhD, but there are really few people who used to get into core and do the PhD during that time. So something might have motivated you to pursue the PhD.
Yeah, I did my PhD in 2008. Well, it is not that late, that early also.
Yeah,
Yeah, I was always, kind of stubborn into doing things which people say are tough. And biochemistry was one such life science field which every student used to say is very tough. We are not going to do. And
yeah, yeah,
you must be knowing biochemistry, how this is, that Leninger grunt, one has to go through it. So when I joined my university, I explored and I learned that everybody in the university, whether it's a zoology, botany, chemistry, or it's the veterinary sciences, it's the home sciences, everybody has to take this particular course. That was so mandatory in every field. And everybody used to cry for Leninger, the cycles, the biochemical cycle. So, but it interested me. I always was happy doing things which are little tough and little different from other things. That's I enjoy.
I think I still remember when I used to study biochemistry. I think students would relate.
Failures, a lot of failures are there in biochemistry.
that that's true.
Tough to
So
life sciences. Yeah.
so of course we would give you some time, to present your research. But I would also like to know about, your early research on capsaicin. That how what exactly you worked on it, and how did you shape that project ahead?
So in our university, that was PAU, it is almost one of the third best university in agriculture universities because they export good quantity of good, varieties of the seeds for
Mhm.
the agriculture purpose. So my there the MSc is also different. It's not like the regular MSc which we do for 2 years. It's like when you join the university, your guide is allotted to you, and your research starts there. So, and it's not like 2 years are gone and your MSc is over. It's like your dissertation starts, and when your dissertation is final, it's done, approved by your supervisor, then only you'll get your degree. So for me, it took me 3 years. A lot of my batchmates, we were 8 or 9 in number. It was three for some, for 3.5 for some. It's not like regular 2 years. So it's like, well, in the beginning, it was not my choice to take my supervisor. It was whatever was allotted to me. But yeah, the field was definitely, I was interested in that.
Mhm.
That was my minor was biotechnology, biotech with microbiology. So we were supposed to do some tissue culturing. So in the initial, like, nowadays what I see in students, the difference, I don't know, I'm not comparing them with my level of work, but yes, they are like what you give them, yes, they will do only that. But when you enter a research lab, you need to do every work which is being done in that lab or department, whatever, so that you get accustomed to every instrument, every kind of experiment going on there. And so that when you finally do your work, it's going to take, weeks or maybe months. So when I started my research in MSc, I remember we used to go to the biotech department and we used to make medias for everyone. We couldn't say no. We were not allowed to say no. We had this, big autoclaves, about 500 liters autoclave machineries were there. And we were, whosoever said to us, prepare this media, that media. So we will go to the media room, we'll prepare the media, say, inoculate our tissue, explant. Okay. So we were getting our expertise in, doing tissue culturing of so many plants, not only my plant, which my supervisor allotted to me. So in that 6 months of optimization of preparing so many media, and using different kind of explant, and how to do the exenic treatment for different plants, and how to monitor their growth in different time periods, how to do the callus culture. All this was learned in 6 months. And then thereafter, when we started working on our own explant, that was capsicum annum. There were two different varieties, so on which we worked. There were the hottest varieties, Punjab Lal, and there was one more variety I forgot. So that was kind of to increase the capsaicin content, which has its medicinal value, especially in rheumatoid arthritis. So its medicinal value was studied from the callus cultures. So thereafter in PhD, because since I worked in tissue culturing, I was again in the tissue culture lab. And here again, whatever the other, professors in the nearby lab, they used to, because as she said, I was project fellow in the NOVAD project. So being at some, though the post was small, so everybody used to say, prepare this, do that. So we used to do that. Besides doing your own work, we never used to say no. जेट्रोफा का लगा दिया, ये मीडिया बना दो, बना दिया, ये बनाना का लगा दो, लिली को लगा दो, you do everything, and then you do your own thing. So these all things, they teach you that not every explant, not every tissue culturing is same. So you have to use your own brain, you have to research papers, and that is very much necessary before pumping onto the table, or your wet lab table. It is very necessary to read the SOP of the lab, and then not only the SOP, because every student has its own way of doing the work. So besides reading the SOP, you should always read the research papers and do your own optimization, which is very much necessary. Your handling errors are different. So I still remember, my supervisor was very nice fellow, and he never used to scold us, never. And but his silence was great scolding for us. So being new to the lab, we were like chitto chatting, also doing work also. So it was almost 3 months. I remember I was doing my protein estimation and doing SDS page, and the results were not coming. So protein was not precipitating somehow. So my supervisor had been to US, and then when he returned, he very gently asked, "So what about your results?" I said, smiling. So protein is not precipitating. I don't know why. It's been 3 months. So he said, it's a serious issue. Just very calmly he said, but his eyes I couldn't forget till now. And on the top of that, I'm saying to him, sir, today is my train for Jalandhar, so I'm going to Jalandhar. He said, cool. And I went home. I realized that he didn't say a word, like what that normal tone was not there. So in the morning, 7 o'clock, I was back in my lab doing my experiment. He came around 7:30 and he said, "You didn't go?" I said, "No, sir. I canceled my tickets. I'm doing my work." I realized that he was serious. He was not smiling, and he was angry with me for not being serious. So thereafter, I was on my own and started my work seriously. And within 15 days, I was onto my bands on the SDLs page. I showed him. Then because optimization was done, you have to troubleshoot things. I was going as per the SOP without changing the parameters. So it's just like how you change your parameters, how you just play with your things that turn on many times. So then I remember I had my, they were satellite markers for DNA, and that result was also not coming. So it's just how when you get serious onto your job, very serious, very, very serious, then only the things that turn on. Little distraction, it will not give you the things, whatever you want in your life. So the pursuit of happiness has to be total. It should be 200%, not only 100%. So the results they came for that also. Now the results they started coming. So I remember my supervisor's wife was also professor, so she was on the top floor, and her results for the DNA for her own, things were not coming. So she came to me and she said, "This is not done, Varna. Our results are not coming. You have to bring our results also." So the point is never to say no to anyone, especially when it comes to learning. So I said, "Yes, ma'am, why not?" So I did her also, and her results also came. So it's not like I'm being overburdened by people saying so many things to me. It's I'm only learning. The lesson is that you are only learning when your supervisor or other teachers or other people, they give you work because they rely that you can do it. So do it. It adds on to your own learning, onto your own growth.
I think that's really beautiful idea of not saying no, at least in the starting phase when you
Oh,
are
that's
learning.
a habit. Still now, I don't say no. Any opportunity which bangs on to me, I cannot say no. I have to work till 5 in the morning, till 4 in the morning. I will take it. I will do it. Unless and until it's a major health problem, that has become a habit. I don't say no to any work which has been allotted to me. I don't think so. I have said no. Even to the reviews which come onto my emails, I have never said no. I have almost done 90% of the reviews which did come on.
I think that's fantastic, and today we have really, I would say, even me too, including me. I mean, the students have become really, like, would I say, narrow-minded a little bit. That they only consider, okay, I'm working on this project, so I'll only work on this project because, I mean, but this I would say there's a really really good piece of motivation.
We have a lot of energy, we don't use it. you know uh, Again, my supervisor used to say, early man, he was slim, he was hardcore. We don't need food 3 times a day. He
Yeah,
used to eat food for a day, then he will not get food for the second day, probably he'll get food for the third day. So we can also survive, we can also do a lot of work. And probably we have, like for example, in our job, we have to just to work on a computer. I'm not saying don't maintain your work life balance. That is also meant, you should maintain, and that can be maintained. It is all management. It's not like I don't enjoy. I also do enjoy.
yeah, that's true. I think that's really really true. Okay, so since you also received the award from Dr. APJ Abdul Kalam, would have definitely significantly stayed important for you in your career. So maybe can you tell that what, like, like how did they choose you, and also how did you feel when you were honored by the President of India at that time?
So, as this is almost linked to my previous answer of not saying no, uh,
Mhm.
And this was the experiment which I was doing in the other lab. My work was distributed in different places, like in Kurukshetra lab, in Hisar lab, in Karnal lab. 3 stations I had, and I was residing somewhere else. So I had almost to get up, take my bus as 4:25 a.m. was my bus, which I have to take up every morning. So getting up at 3:30, get ready my daughter, the school dress, and then she was sleeping again back so that she is ready at 3 o'clock. She was ready with her ponies and her dress. And 4:25 we used to leave because of work on 3 station. So this experiment I was doing in Kurukshetra lab, and I remember this was a chromatography experiment in which I had to purify certain enzymes, acid invertase and neutral invertase enzymes were there. So the experiment has to go till the Km value, which is the Michaelis-Menten equation. You very well know that is Km. So somehow the results didn't went to the end because my sample got deteriorated in the cryocan for some reason. So I told my supervisor that my sample has got deteriorated and have to wait for next year for the sample to get the sample. So so Professor Dhawan was my co-supervisor, kind of.
Mhm.
So my main supervisor said, "Yeah, you can wait, but there was a thing that okay, you have already done 10 experiments, it's fine. So you submit your thesis." So, but this experiment, whose Michaelis-Menten equation was not calculated, which I had done, though the purity level was checked, all these things was.
Mhm.
So this was not added into my thesis.
Mhm.
Because yeah, it was like the final goal is not achieved, so we will not add it in your thesis. I said, okay. Uh, so 9 experiments were added. But Professor Dhawan was another level professor. I will not say that Noelder is definitely it's my work, but it's the effort of my supervisor. So every
Mhm.
supervisor, every good supervisor does so. I have not seen any good supervisor except mine, so I can't say they must be existing. This work, which was not published in my thesis, was granted this Noelder award. So mean to say that no work is getting wasted. No, none of your research gets wasted if you think that मैंने उसका काम करा या whatever I did, some other thing got wasted. No, if you did it with dedication, nothing is wasted. So that work he presented in such a nice way in a paper that was selected for the Noelder Gold Medal Award. And I didn't have this information that this was selected and all those things because obviously he was intimated about that thing. So I remember I was at home and my supervisor called me. So I also have seen that good people or good intelligent people, they have good sense of humor also. So he called me and he said, Vannaji, your research, this is not good, and I think this is not going to get. We will not write paper also on it. Something he demotivated me. So I was like, okay, fine, ठीक है, PhD students को क्या होता है, जो कह दिया supervisor ने ठीक है, हाँ ठीक है, okay, fine. I was no कि हाँ ये करेंगे वो करेंगे ठीक है, whatever at that stage is going on because you are not your own, you are onto your supervisor. I said हाँ ठीक है. So my supervisor wife, she took over the phone because he had demotivated me so much. Said, Vanna ji, your paper has been selected for the Kalam Award. I couldn't believe it. I said, really? She said, yes. Then he took over the phone. He said, yes. And we have to go to Udaipur and Radisson Hotel, and there the ceremony will take place. So it was like, okay, fine. So that was, yeah, cloud 9 moment. I was very happy.
And while receiving the award from Dr. Kalam, how did it?
It was the feeling I cannot explain. Uh, the person was so humble. So I mean, no, I mean there is. Just feel like you are talking to a casual person. So when I was the only female, first of all, and the youngest female in almost there were 14, 15 elderly people. They are sitting at the side who were going to get those awards. So when my number came in, he had this medal uh, ribbon and the medal which has to be put onto my neck, and he said me, "Is it okay if I put it onto you?" I said, "Sir, it would be an honor." Because maybe considering the Hindu facts on all this, he must have asked that question. He said, "Is it okay if I put this onto your neck?" I said, "Sir, it would be an honor, please do so." So that was only conversation. Obviously on stage, what else can you have? But that
Yeah,
is a very down to earth man. We all know him. Yeah, he's really
yeah,
a wild man.
yeah. He was like my role model, and I've always wanted to meet him in
Yeah.
person, maybe one interaction, but I think that was not on the books.
So, yeah, we are all lucky that he was our president.
That's absolutely true. I think India saw a really great, one of the very great president.
Known worldwide. Yeah.
Yeah. So since currently your research laboratory is focused on diatoms and biofuels and nanotechnology, so maybe you can give like a very small brief on what exactly you do currently on diatoms, and how do you see their potentials in biotechnology like in upcoming era?
Okay. So I will like to club the diatoms because with the forensic and the nanobot. First of all,
Okay.
I call them nanobots because of their
Uh-huh.
nanopores which are there in the diatoms. So we all know that diatoms, they are the macroalgae which are found in water, uh,
Mhm.
invisible. And it has a lot of forensic application. As I said, I was in the forensic lab, so
Mhm.
we
Yeah.
work on diatoms. So first of all, let me tell you what these diatoms do in the forensic lab.
Mhm.
Uh,
Yeah. Yeah.
yeah. So these diatoms, as I said, these are the microalgae, and they are found in every water, every open water, provided it's open. It's not a tank which is closed. So as you know, it's a plant. Microalgae is a plant, so it is
Mm,
open.
mm,
Oh, so it is photosynthetic. So they are found in every open water. So now,
mhm,
how it is related to forensic is that the when a body is found in or near the water,
mhm,
when it's a case of drowning. So when it's a case of the drowning, the test which comes in the forensic science is the diatom test. And
mhm,
diatom test is considered as a golden standard to detect whether the death was antemortem drowning or postmortem drowning.
mm,
So antemortem, as you know, when the person is alive when in water and dying.
mhm.
Postmortem, the person is dead and somebody has
Yeah.
thrown the body into water to
Mm,
simulate it to be a case of drowning. Now, as a science student, we know that when we are breathing oxygen, this oxygen is being carried via pulmonary capillaries to the from the lungs to the heart, right, via pulmonary vein, and is carrying the oxygenated blood. So the pure blood or the oxygenated blood is being carried. So now, when in water through the nostrils, not only the oxygen is going, but the water is also going. So now this water mixed with oxygen or the air is now entering the pulmonary capillaries and the pulmonary vein. Now, since this water is having the diatoms, it also enters. It also enters our heart. So if it enters our lungs, it enters our heart. But we are not taking lungs as the correct sample for diatom examination. Why? I'll tell you. But we are taking heart as a correct sample for diatom examination because diatoms are entering via pulmonary capillaries and vein to the left side of the heart. And from the left side of the heart, they are being carried to the different body organs. And the next best sample for the diatom examination in case of body found in water is the femur bone marrow, that is a thigh bone, because it is protected from any contamination. Now, we take this sample from the heart or the femur bone marrow and we do the analysis and we found the diatoms. So if the diatoms are there, the death is antemortem. Now, once a person is dead, so he's not respiring, he's not inhaling. So there are, there are some muscles which will close this inhalation inside. Sphincter muscles are there. So the oxygen is not going inside. So oxygen is not going, so water is also not entering. So the sphincter muscles, they have closed this gap. So the water is also not going, so diatoms are also not going. So the detection of the diatoms in the heart and the femur bone marrow is negative. So that's how you detect that this is a case of post-mortem drowning and this is a case of antemortem drowning. You're getting my point. I can show you the slides also, but once I come onto the slides.
Yeah.
Yeah. So, yeah, you want to.
So I had one doubt. So since you mentioned that after the death, the sphincter muscles they get blocked and nothing can enter inside through your respiratory tract. But what about entering through your
It's common. It's not going anywhere. It's not going. So we are not.
uh,
Oh, okay. So from mouth it will go to the stomach only, na?
yeah, that's
So we are
I
taking
thought that.
sample. So we are not going. We are not taking the stomach sample. So stomach sample is a control sample you can take. If you want to take,
Mhm.
you can take the stomach sample as a control sample. Although the control sample we take, 1 liter of the water is collected from where the body is recovered. The IU, the
Mhm.
investigating officer, he collects 1 liter of the water. It's mandatory. So while selecting the body from the water, it is mandatory to collect 1 liter from where the body is recovered. Also, if the water is in the intestine, that is also a control sample. So
Mm.
the nasal passage is closed. The nasal passage is carrying the oxygen to the lungs.
Mm.
The mouth
Mm.
connection is via esophagus to the stomach. So the connections
Mm.
are different.
Mm. Mm.
So
I
the connection
understood.
is different.
Yeah, yeah,
So
yeah.
I can explain, but that's going to take long for the particular class only.
I think it's a little bit difficult concept, but maybe but you were developing it really well. I could follow. I mean, I think people would also follow it.
Yeah. I almost did many cases in Diatoms. Although we were doing other cases like the rape cases, murder cases, hanging cases, railway accident, so many other cases also was a part of the biology lab. But Diatom examination slowly and slowly became so much part of me that whenever there was a case of national importance, the director used to hand over to me, and not only the director, but then the request used to come from other laboratories or other officers that we need to get this case done by Dr. Vandana. I remember after Nirbhaya case in 2012 December, there was a case in April 2013 of Bhandara rape and murder case. So I'll not say the name of the SP, but she called me up and she said that we want this case to get examined by you. So I told her that, see, I cannot say that I want to examine this case because that's a biasness. Why will I? There are other people who can do the case. So I said, if you kindly talk to the director, whomsoever he will log the case, that would be done. And everybody does with full dedication. That is not an issue. But anyhow, she must have talked, and the case was allotted to me only. So Bhandara rape and murder case of three sisters I did. Then I did Shofia rape and murder case in Jammu and Kashmir. Then there was a headless dead body case of Panipat. So like this, many cases of national importance were done.
So when you try to solve these cases as a biologist, I remember I got an admission into the forensic science course from the National Forensic Institute, which is run by Central Bureau of Investigation. So I was just applying and I was not sure whether I will get it there or not, and I was just applying in a way that, okay, I'm looking for masters and something new. And I really love the forensics and
Right.
the mystery.
The science is really, I mean, you are working for some cause actually. I like this science because doing science is good, like we all do, writing research papers, but doing forensic science, it gives me a sense of satisfaction. You are doing something for the society for
Yeah.
practical usage. So we do
Okay.
a lot of science and we say it has practical application, but to be very true, it is going to take much time to bring those things actually into practical applications. We keep on saying this is a pilot scale, this is a lab scale, this is a benchtop thing. So, but
Hmm.
when I do this science, forensic science, I really feel contented a lot more than what I do other sciences, which also is a part of my core subject, because it gives, a sense of satisfaction that somebody has been given justice or I have done something for the society.
Yeah.
So I love this science for that reason.
I think that's really fantastic. And do you ever get like chills when you investigate the
Yeah,
case?
lot of time, lot of time. And there was a time, there were times like, when we used to, so, there are many cases. I remember when I was in the lab, the average 150 rape cases were reported every month. And we also knew while doing the cases, while reading their case histories and sometimes the PMR or the MLR, that this is a genuine case or this is a fake case. But we cannot say because we don't know which case might be a true case. So we have to do every case very genuinely.
Mhm.
Uh,
Mhm.
but yeah, I used to get chills when, especially when there were heinous crime cases, like when there was a rape and murder of 4-year-old child, or whether it's a girl or it's a boy, a case of sodomy or something. So that moment, I was really in my, like I used to, like, my daughter, has she come from school or not? So that night I couldn't sleep. Those nights were really chill because you have your own children. So, and especially when you have nowadays, when you see the Kolkata case, I'm not going to discuss on that, but yes, as a mother, you really feel you're a little worried, like, where are we going as a society? Definitely. Why these cases are not ending up? What is the mindset of the people? Why they
Yeah,
are doing this? So,
yeah.
yeah.
I think absolutely that is something which is
So,
deteriorating our humanity, and also how international market is also looking today towards to this thing. I think uh, everyone gets chills.
yeah.
And did you, I mean, I don't want to get into serious troubles, but still, like, do you get threatened for getting manu like to manipulate the data? Okay, let's say this, or you do this, then you'll get this or that.
There's a very good answer to this. So my husband, who is also in the forensic lab, he just taught me one thing. By the way, he is a core forensic guy. He did his
Uh-huh.
BSc forensic, and he is a gold medalist from this university from where I'm teaching right now. So
Mm-hmm.
he just told me one thing, and that was never pick an unknown number.
Ah,
Whatever
okay.
numbers I have saved, I'll pick only
Mm-hmm.
those. So,
Mm-hmm.
I shouldn't say many things officially, because you have to maintain the decorum of your case file. So,
Yes.
even if some known person comes to my room cabin and asks me, Madam, has this case been reported? My answer was very, uncanny and sarcastic. I said, there's so many cases. How will I remember every case? I don't know. No, no, you just look on this case. I say, I have a little bit of idea. So you can answer if you want to say polite no. So people understand that she does not want to say. I remember, also remember, there was, it all depends upon you, because when you're in the lab, you have to take oath, and that is
Mm-hmm.
not just taking the oath. You have to, stand on that.
Mm-hmm.
So there was a case on which there were seals were not there, and
Mm-hmm.
I said I will not open this case because the seals are not intact on the parcel. So I was asked by the senior officer that this is a simple case, just do it. There is इसमें कुछ लिया दिया थोड़ी ना है, just do it. Uh, I returned the file back on the table, and I said, Sir, even if the Director General of the police will say to me to do this case, I will not do it because it doesn't have the seals. I will not do it. So you have
Mm-hmm.
to be very clear on many things. And yeah, when you have, I remember when I did Shofia rape and murder case, that was in that for that you have to go for your expert testimony in Jammu. And since it was a very high profile case, so a lot of protests were there. When my report was submitted, there was a Jammu Bandh that day. So I had told upfront to the AIMS and the CBI officers that, see, I am reporting this. When I reported, I didn't know that this is a high profile case anyway. Because when I joined the lab, I was told to come immediately and do this case till it finishes. So I didn't know that media was there. Media was standing outside. I thought maybe some case must be there, but I didn't know the case I'm doing is for which the media is standing outside. So the case was done on the weekend, and I did the case till 3 in the midnight, and I did it and reported it. And the director was also sitting in the lab. I said, "So these are the results." For them it was good because they matched with the post-mortem reporting of the doctor, so they were fine. They were happy that yeah, it matched to their results. And then they said, you have to come to Jammu. Uh, just a few years back, I had lost my brother. He was in the army, and he was killed by the militants. So my parents were a little afraid for me to go to Jammu. I said, "Sir, I will not go to Jammu." So he said that, "Okay, you make a PowerPoint presentation for us of your report." And obviously, my report was sealed and given to them. So I made a PowerPoint presentation, and for the explanation in the court. So yeah, I didn't go. So but I went back to my home, and then when I came in the morning, so everybody in the lab said, "Madam, Jammu band karwa diya aapne." I said, "Oh, what happened?" I said, "This is okay. You just have to be cool. You just have to be normal, because you don't want to be, things to get exaggerated. But yeah, that's a part of job, and aisa nahi hai, you are protected, you are safe. This is not the. It's okay. You take care of yourself also many times."
Yeah. I still have a dream to work with forensic. I think I don't know, but I have not seen many biologists working in this area, especially the diatom test. I personally had never heard about it, so maybe it would be popular in the field. But
I mean, it is, at least it is giving justice to many.
yeah, absolutely, that is more important.
That is more important for me. That I am happy to do internally. I'm contented to do things which, when people say, people on their practical, yeah, this thing has happened. Okay, for the sake of science, I am doing science. I am now using my all biochemistry aspect on working on that and nanotechnology for biofuel, which is also going to work for the society, but it's taking a little bit more time. So if I had this Indo-French project from Pacific Prague, which also we have done reasonably good work, and we have brought certain medicinal or pharmaceutical, you can say, or nutraceutical products, patented some products from Germany, and German patent we have got. So all these things which will work for the society, I feel good for that, not just publishing papers. Yeah, that also is because in the system you have to balance yourself, but at least 5% you can do something which is good and people recognize it. Yeah, that is for the society. So that is very much necessary to me, at least.
I think that's really beautiful about the work. So since you mentioned about nanotechnology and uh, and how exactly diatom test you do, so maybe you can also discuss the concept in short about the difuel TM, and how the diatoms are currently used a lot in the biofuel technology.
So you see, the crunch of crude oil or the petrol we all are facing, this is something not new. And ever since I started working on the diatoms from the forensic aspect, also I started collaborating with people who work on diatoms. So I met this Professor Richard Gordon. who coined the term in 1984 on datum nanotechnology. So because immediately while I was writing my thesis, I got the job. So I had this lacuna of not doing the postdoc. So because immediately I was commissioned in the government job. So, but I had this online mentor kind of thing, which now I also do the same. So this Professor Richard Gordon, he is a datum expert from University of Manitoba, and he had been with me since 2011. We are together, working till now. So from there, I started working on datum biofuel and exploring the biofuel aspects of the datum. Although it is existing, it's not something new. People were working on it, but for me, it was new. I know, but I always wanted to work with things which I had learned. So what techniques I had learned, the techniques which I had learned was tissue culturing. So I had to use that thereupon. So I then collected samples. I cultured many varieties of the datum. I made a datum atlas for the Haryana state. Then I found some specific datums which were found in specific water body, which not only were the ecological point of view very important, biofuel point of view very important, but also from the forensic point it was very important. When you find a dead body with that particular datum, you can actually have an idea of the place where the death might have taken place, or the body has been drifted, or even the season or the time of the year. So all this has been discussed in that particular book. So, all the crude oil which we use is coming from the beds of the ocean. The fossilized, it is a fossilized plankton, of which the datums, they are the contributing the major fraction of it. So since they are contributing approximately 30% of the crude oil, as per the geologist survey, so that was very important. Why we are not, having plants where the datums are being used for getting the crude oil. Now, while researching on it, we obviously have found that their silica wall, uh, which because of the silica wall, they had a role in the datum, because that was not getting degraded in our systems. Silica is the hardest material. That's why their fragments are remaining in our system, not getting degraded. So their silica wall is creating hindrance for the oil to come out easily. So obviously the point was to lower the cost. Otherwise, you can cultivate datums, but if the cost of extracting the crude oil is high, then what is the point of doing all those stuff? So the logic was to harvest the oil from the datums, not to kill the datums. It's just like milking the cow every morning for the milk, and not killing the cow and getting all the milk at one go. So that hypothesis we started working on, and we then developed many techniques how we will just fracture the cell wall of the datum, and we just harvest the datum, the crude oil, and on feeding the more of the media, the walls they heal up again, and the cells are viable. So it's like milking of the oil from the datum. So for this, we worked on many techniques like electrical shock techniques, osmotic techniques. So for this, we made many kind of fuel cells, like ice solar cells. We made perovskite cells. We made microbial fuel cells. So all these they started coming on to aspects, and this biofuel from the datum was known as a diafuel. We coined the term diafuel. So yeah, then similarly we found another microalga which was now not only the crude oil, but also it is rich in one of the pigment that is the fucoxanthin, which is having the antioxidant properties. And obviously the crude oil in it just not only having the this locomotory properties, the biofuel, but also it's rich in many good fatty acids like DHA, EPA. So all these are good for our health, right? So the fish oil which we take is rich in DHA, and the fish feeds on the diatoms. Its main feeding food is diatoms. So many places in South I have seen they cultivate diatom as a fish food. So actually, the fish oil we eat, it is one of the ingredients they get it from diatoms. So, and the fucoxanthin, that is a pigment, it is rich in antioxidant properties. So simultaneously, we worked on one more potential alga, that is a hematococcus. They both are robust microalga, and hematococcus also has thick cell wall, very thick cell wall. Both diatom and hematococcus, they have this common problem of thick cell wall and high potential, pigments in them. So this
Mm-hmm.
hematococcus has astaxanthin as one of the very strongest pigment, which is having 6,000 times more antioxidant properties than the vitamin C and vitamin E. So you can
Mm-hmm.
very well imagine how strong antioxidant properties it is having. So that is the reason it is also known as the king of all the existing ketocarotenoids. So on this basis, we have made certain file products in the commercial market, so which were granted German patent, and we have also used the diatom dust to make the diatom fingerprinting powder. So some practical applications. Then
Mm-hmm.
because the cultivation of the diatom is little tough in the climate like ours, because it needs a temperature of 18 to 22 degrees Celsius. So we
Mm-hmm.
need to, and obviously, one more technology which we developed was the bubble farming. It obviously needed the cheapest way of cultivating the diatoms. So now in the open photobioreactors, what we do, the water evaporates and the contamination also results when we cultivate the microalgae. So, and
Mm-hmm.
in the closed photobioreactor, the cost is very high. We have to replace the batteries after every 5 years. So the cost of photobioreactor is very expensive. So we need to develop something very economic. So what we did, we used the discarded bubble wraps, which are come in packing material. Uh, so the air pillows. So we cultivated first we tested all this stuff. We cultivated the datum in the waste plastic wraps, because there was, and it was found that the low density polypropylene, it allowed the exchange of the gases. So then we used this a little higher scale, and it was found that yeah, bubble farming actually worked where there is no, in our lab. I have bubble farming since 3 years. The bag is not changed. There is no loss of water. Can you imagine? There is no loss of water. There is no contamination. So such kind of things, and if you have such kind of diatoms which are self-oozing, which we have discovered one self-oozing property of the diatom, which self-oozes the oil, which was found in one of the sites in Haryana. So this datum, it oozes oil naturally. So once you have optimized a system where there is no water loss, there is no contamination, the system is cheap. Uh, Obviously, it can be taken to another level. Yeah.
That's really something new. To honestly, today's whole talk is super new for me, because I have never heard about anything, because always I was in working in the molecular biology and human medicine side. So this is completely a new aspect for looking into research, I would say. That's really good. And I had a quick one curious question, that are diatoms edible? Do you have food grade level diatoms?
Yeah, we have food grade level diatoms, but yeah, they are poisonous diatom also, which secrete.
Ah.
Yeah.
Harmful oils or something like that.
Yeah, I mean, this one, I guess, not all.
I see. So maybe you can use it like kasuri methi, and you can put it on your.
You cannot do that because it is rich in silica. So you cannot eat.
Okay. Cannot digest it. I see.
You cannot eat silica every time. You can use
I
their
see.
contents
Yeah, absolutely. Something like oil you added from
or
above.
pigments and
Yeah,
oil. Use.
absolutely. Absolutely. So I think, like, I would like to ask, one or two, like, one last question, and then you can, we can have your presentation, and then we can take up the audience questions as well. So we ask this to each and every mentor, or each and every scientist who come to the SciComm synapses, that, like, as an accomplished scientist, educator, and also a mentor, what advice would you like to give to the young researchers, I would say, let's say, master students or PhD students? So what advice would you like to give from your journey till
My only advice is that be motivated, do whatever you think you do. You just need not lose your motivation. And without hard work, there are no shortcuts. If you want to attain something, hard work is very much essential. You have to keep yourself motivated. And actually, there is no new lesson I would like to give. Without hard work in any field, there is no, good results. Hard
Yeah,
work is
that's true.
really you have, and you have to maintain it. Okay, I have done my PhD, now I need not do anything. No, that's not going to work. Okay, I have done my MSc, now I have got a job, or now I need not do that. No, you have to do hard work. That is what humans are for on this planet, and you have to maintain it. You have to maintain. It's not like you've got a job now, 9 to 5. You go or you don't go, or you go 11 to 4 and you're back. Then don't say why that person is better than me. I have more. Maybe you have more higher education than that person. Lately, I met a guy. I had been to IIT Bombay for some work, and in the lift, I met this boy. He had this some model in his hand, and he was explaining to someone. And so I was curious to ask, "What's this?" I said, "This is my startup." I say, "Means?" Yeah, yeah. I have. He explains so many things. I was jumbled on, but yet what I got the message, the hunger in his eyes, I couldn't explain to you. He was just 12 and he said, I had no time to take higher education. I'm onto my project and I have to bring it to the pilot scale. It's worked on the smaller scale. I have to bring
Oh,
pilot scale.
wow.
I talked to myself, this is Elon Musk, future Elon Musk.
Yeah.
He was, I hardly see, motivation or that, hunger in the eyes of students to do something. They just, okay, the guide will say, or the mentor will say, or the teacher will say, okay, kar lenge, ho jayega. No, within yourself, I will do it. Okay,
Yeah.
this mentor has said to me, do like this. What I will add flavor on it. Let's do this. Without saying when you do the things, you do it for yourself.
Mm,
And then that speaks. That results are seen by everyone.
absolutely. I think, and also one more thing I would like to never say no. That's something I would learn from this
And
podcast.
always
Absolutely.
ask. And always ask. If you are in trouble, always ask also.
Always ask. Always ask. Yeah. Never
Never
say no and always ask.
presume things. First, never say no, always ask and read. Nowadays, everything is free.
Yeah.
Internet, everything is free. So you cannot say that I don't have. You just have to manage your time.
That's absolutely true. Maybe we can now proceed ahead for your short presentation.
Oh, it's not short. I'll skip because well, I have discussed many things orally also.
Okay.
But yeah, the pictorial demonstration would be good.
Yeah. Okay. 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 Maybe 5 minutes or something like that. You can
Yeah.
definitely. I would definitely love to visit your lab when I come to India. I think I'm coming in October. I want to see the forensic bio, biochemistry or forensic laboratories.
Wow.
How they.
So
I know I don't have permissions to work, of course, but
no, no,
maybe.
no, no. This is the research lab. When we were in the forensic lab, there you have to definitely seek permission to enter. Well, that also it can be managed. But yeah, in the research lab, definitely there is, as such, there is no permission is required. You can always.
Sure, I would be quite happy.
So okay, this is I just prepared. So also I'm having some research projects which I had worked from various agencies. Like the
Mhm,
recent one which is going on is the Indo-French project, and
mhm.
which is on the, as I told you, the pigments from this algae. Then there was a DBT project for PCR markers for the diatom to link depth to drowning.
Hmm, that,
Then
that, that,
uh,
that's something.
yeah, then there was a UGC's project on molecular identification of some forensically important insects. Then there was
Hmm.
a DST nanomission project on construction of the diatom solar panels for bios.
Solar panel.
Yeah, yeah, yeah.
Interesting.
Yeah, I'll discuss if we get time. Then this was a Erasmus funding. Then there was a GIAN project from MHRD. Then the project from INUP IIT Bombay for some microfluidics. Then NCPRE IIT Bombay. again for some fabrication of devices. Then there was MOU and the DST project, which I was discussing with you, the first project which I got
Yeah.
during my thing. Yeah. So diatoms, they look something like this under the scanning electron microscope, and they are not something new. Since the Jurassic period, they have been there, and they are crude oil depositors, as I told you. And these are some old diatoms, old time diatoms, Oligocene era diatoms, or the Omaru
Mhm.
diatoms, we call them. So, as popularly known as the living jewels, these diatoms, you can see the beautiful SEM
Uh,
images.
we cannot see your screen. Maybe can you
Moving.
present it? No.
Okay, I thought. uh, So, okay, I'll share it again.
I wanted to see the electron microscopic
Can't share
diagram.
your screen. I don't know why it's coming. Um,
Yeah, I think you can. Maybe go for the entire screen or something like that.
I don't know. I'm on the this Firefox.
Okay. Firefox. I am little bit. I am not a technical, not good with technical person, but
Can't share. Firefox might not have screen recording permission on your computer.
uh,
It doesn't allow.
Can you try once?
Okay.
oops, I don't know what is happening. Hmm, why is my sharing? Just a second, Dr. Vandana.
It's
I
not
think
allowing.
I will just rejoin in a second.
Okay.
Ma'am, are you able to share your screen or means is it some dialog box coming up
It's
when you are presenting?
it's saying only share screen with the sites you trust. Sharing can allow can't share your Firefox might screen recording.
Okay,
So,
okay.
no, not share screen. You have to
Uh,
click on present now.
Yeah, yeah, I know presenting those some. Okay, maybe join the other.
Familiar, maybe you can mute a little bit. I
I'll
think
join with
it.
other system maybe. Yeah, I'm with Mac. Mac is creating two problem. I think.
Okay. Or maybe you can email me and some, or to the community. They can do the screen sharing.
It's a lot of slides. You will not know.
I
Uh,
see.
yeah, just a minute. Hello, can you hear me?
Yes,
Yes, ma'am.
absolutely.
I'm sharing it now. Maybe it will work now. Is it visible?
Yes, it is visible.
Okay. It's visible?
Yeah, yeah, absolutely.
Okay, fine. Take care. So I was telling about all this stuff, the diatoms, and these. Can you see now?
Yes, yes, we can
Yeah.
see.
So if you have not seen it earlier, so these were the different projects I was discussing with you from the DST, UGC, DBT, CIFIPRA, and NCRE, NCPRE, IIT Bombay, INUP, IIT Bombay, GAN. And these are the diatoms. These are the old Umaru diatoms, and these are the some scanning electron microscope images of the diatoms. And we say them as also as a gold brown microalga, since they are two walled, and definitely they are the eukaryotic plants with they fit like the soap case. And regarding the forensic application, as I told you, that any body found in or near the water, for that datum test is very important. And drowning is not necessarily a body should be in a pond, river, lake. It can be in a mug of water also. It's just like blocking the nostrils, where only the water will enter, provided that water, in diagnosing diatoms, the water should have diatoms. So if you take the distillery water, drinking water, we cannot find and if the drowning is with that water, we cannot find diatoms in that. So it has to be the water where the diatoms are there, and it has to be some open lakes or puddles or rivers or oceans. So this is the schematic if you would like to understand this. This is a case of live entry. You all can see the slides.
Yes, yes, absolutely.
So this you can see over here. This is a live entry of a person, and these are the diatoms which are microscopic, not visible to the naked eye. So when in a live entry in the water via the pulmonary circulation, they will enter the lungs and they will enter the heart and then to the femur bone marrow. So these two samples are the best samples for the diatom examination. The tests are positive, right? And this is, you can see the person is dumping the already dead person into the water. Here the diatoms are not found in the heart and the bone marrow, but you can see few diatoms are found in the lungs. This is because the lungs, they are prone to give the false positive test, because when the body is in water, there is a decomposition of the body. There is a gnawing effect where the body is being eaten by the animals also. So the percolation of the the water body, the water from the water body can occur to the directly to the lungs, which can give the false positive test. So that's why the lungs are not the right sample for the datum examination. When I joined the lab, the two samples which we examined for the datum examination, two regular sample in every datum case, was the sternum bone, that is the chest bone, and the clavicle bone. And these two bones, they are the wrong samples for the datum examination. So we took a drive in which we wrote to the medical officers and PGIs and the IOs that please don't bring us the clavicle bone or the sternum bone, bring us the heart or the femur bone marrow. So things this took a tough turn to change, because taking out heart and the thigh bone, it was a discomfort for the families of the deceased, because they don't want they for them to explain, us to explain, the IOs and the DSPs and the medical officers, and for them to explain the relatives that if they want justice, this is the right sample and we need to take it out. So, and now slowly and slowly, I think things have changed, and they are getting the right sample for the datum examination. So this is a little more of the science. I will skip on to this because not everybody would be interested. So the signs of the drowning, they are like the regular froth is coming from the mouth and the nostrils. Then there would be clenching of the weaves. The lungs would be swollen and voluminous, and there would be indentation of the ribs of the lungs. So that's why being a forensic scientist does not only means doing a particular analysis, which I always say to my student that we are not scientific laborers. You also need to because everything is regarding human, so you need to understand the human system also, because if you just know the some chemical examination or physical examination and you're not able to understand what the doctor has written in his post-mortem report or medical legal report, you cannot bring justice to that particular case examination, because you don't know whether your test needs a repetition or not, or whether you have taken the right sample or not. But once you understand the technical language written by the doctors in their PMR and the MLR, you will be able to judge, yeah, yeah, I need to repeat the test, or I have taken the wrong sample, I have to take it from other things, whatever things are there. So that's why, because you are circled around the human being, you need to understand each and every anatomy of the human system also. This is the froth from the nose and the mouth. So these are the just symptoms which I'm just skipping this slide. And this is a method which we are using, that is the acid digestion method. This is the femur bone and the water sample as a control sample. Then there's a digestion and the microscopy, and we see the diatoms over here in the microscope. So as I told you, that is this is the golden standard test for detecting the postmortem and the antemortem drowning. Now, while all this was going on, the method which we use, that is the acid digestion method, it generated a lot of nitrous fumes, which was not good for the staff which worked. So I generated one protocol, that was a DNA protocol for diagnosing the diatoms in the drowned victims. So I tested on the mouse, and we standardized or optimized certain primers which were helpful in detecting the diatoms in the drowned mice. Sorry for those mice which we killed. Uh, this was published. This is a detailed study where we use the markers which was specific for the ribulose 1,5 bisphosphate, which is a photosynthetic marker, which obviously the responsible for photosynthesis. So because humans, they don't have plant genes, so how you can differentiate the humans from the plants? Although both diatoms and humans are eukaryotes, so you have to select the chloroplast based genes because of the limited homology between the human and the plant genes. So this was published in the Journal of Electrophoresis, and later it was also published as one of the cover page journal for this particular journal, Electrophoresis, this particular findings. And I still remember the reviewers, they wrote that this is a very novel work. Well, so then we studied different diatoms, and then we marked their cross lineage. So this is a little bit of mol bio, and this is certain cases which I was discussing. One of the case of homicide, means the person was killed and then put in water to simulate as a case of drowning. This was a negative examination. The diatoms were not found. There was no frothing. There was no clenching of the hand. A lot of things that need to be discussed. I'm just skipping. And this was a case of national importance, that is a Sofia rape and murder case, which was in collaboration with CBI and the AIMS. And the biological sample, the bodies were actually, these were exhumed from the because of the religion, the bodies were exhumed, and the postmortem samples were taken out, and the examination was done, and which matched with that of the water sample, which was the control water sample. And then after Nirbhaya, this was the Bhandara rape and murder case of three little girls from Maharashtra. So this was presumed the case of rape, but this was not a case of rape. This was a case of drowning. So yeah, any time for all this discussion, I cannot say in few words, so I'm just skipping all this. So the correct samples definitely need to be the blood laden tissue from the left chamber, the femur, tibia, humerus, or the distant organs like the brain and the kidney. Definitely not lungs, not sternum, not clavicle, and obviously the water sample from where the body has been recovered. So yes, they are very good tool, a golden standard tool for diagnosing this drowning, and this was also in collaboration with my DST and DBT project. So as I said, I clubbed the slides. So, and since they are made up of silica, so they are natural silica, which are having a lot of functional groups, and because of their functional groups, they have this ability to get doped or linked with many linkers, so that they have their great role in uh, the therapeutics and pharmaceutical industry also. And because of the silica, they have their great role in optics and the photonic industry also. So it's a organic silica, natural silica, compared to the silica which is so expensive. So there's the natural sources, and via FTIR we saw that since there are 20,000 different type of the diatoms, and via the functional groups we grouped them into four different groups. And how we found that the diatoms which are oval in shape, they had the common functional groups. The diatoms which are elongated in shape, they have their common functional groups, and the cellophane groups were there. So this is all the application which you see then: environmental technologies in the raw material for the industry, in the biomedical, in the drug delivery, in the nanoparticle, the biosensors, in the wastewater treatment, the nutraceuticals, for genetic engineering, phytoremediation, river ecology, so many. And the datum solar panels, they are nothing, just what are solar panels? They are the glass panels. So, and the diatom wall is also made up of silica, so that's why we are calling them as the diatom solar panels, because diatom is a living organism inside the glass houses. So that's why we're calling them diatom solar panels. And there, the normal solar panels is giving you electricity when exposed to sunlight, and here they also need the sunlight, but they are giving you the crude oil. So that's where the diatom solar panels, which are giving you the crude oil for running your vehicles, or directly for the consumption purpose also. So all this I have discussed. Then there are a lot of application. As I told you, we had made many different kind of cells, like the dye-sensitized solar cell, lab-on-chip devices, the microbial fuel cell. I'll not go into the detail because it's not going to happen like this. So this explanation I gave you regarding the milking. What we do is the milking of the oil from the diatom. Just like we don't kill the cow, we milk the cow every morning. So that, just like that, we need to devise a technique via which we do the milking of the oil from the diatom. This is the diatom, and we see the milking, and just not destroy or extract. This is extraction. So we need to heal this diatom rather than killing this diatom. So that only can be a very economical way of upscaling these techniques. We have used different techniques like mechanical pressure, doping, pulse electric field, and then we have found this natural losing property in the case of diadesmis confervaceae. This is the TLC, and this is another diatom on mechanical losing. And then these are some AFM microscopy images of the datum, where we have used the gold nanoparticles, and we are trying to see again for the energy purpose and the oil losing purpose. So again, some nanoparticle magnetite, which we have used to harvest the oil from the diatoms using the magnetite nanoparticles. Then we had used TiO to dope. This is a polydimethyl siloxane, which is a hydrophobic material doped with Lufa cylindrica for photocatalytic nanosponge. It is like you are exposing them to the UV light, and how they are oozing the or absorbing and liberating the oil. So it's good for the oil spillage industries. So like this is the plan is like you have a water tank at the house top, and you get water. Now instead of the water you're getting the oil. What about this? So it's cool, like once we have such kind of techniques in our system. So this was a bubble farming. As I told you, the used air pillows which we get in packing material, where they were used for culturing the alga. So it has no water loss, no contamination, and high diafuel, or the biofuel from the diatoms. The water retention, as you can see from the bubble wrap, there was absolutely 0% water loss as compared to the test, which was open sample. So like this, we have devised many. We have used many waste materials for the cultivation, like even the drip bottles were used for the cultivating the algae. And you see, these are the oxygen curtains which we have devised in the labs. So oxygen curtains, because they alga, they are liberating the oxygen. That's why we call them the oxygen curtains. And another point is that they since the alga, they need phosphates and the nitrate as a major component for their growth, and the wastewater is quite rich in both these components. So the waste water, if it is properly pre-treated, it can be a very good material for cultivating the diatoms and other microalga, and that can be economical way of upscaling the cultivation. So for this we had checked many wastewater pollutants for estimating the diatom growth, and we found that the crude sewage water, it on certain dilution level, it gave 10 times elevation in the cell growth and of the diatoms in this particular case. And as I said that they are also acting as an ecological indicator. So one of the diatoms, that is the Gomphonemma pseudo, this is its natural shape, but due to the some heavy metals in that particular water, that is one of the Saraswati Dham in Kurukshetra, Peva, where this water site we collected. So this is the normal shape of this diatom, and because of the heavy metal pollutants in this of the lead and the selenium, it has distorted its shape. You can see there's a distortion, which is the morphological deterioration, which we say. So this is an ecological indication that some heavy metals are there. So yeah, I'll skip on to these slides. So these are the dye-sensitized solar cells and the metabolically engineered diatoms and the lab-on-chip devices which we made at IIT Bombay, and how these act in harvesting the biofuel or the diofuel from the diatoms. And another diatom microbial fuel cell. So you can see the electricity is also generated. Not only the wastewater is being treated, it is being used for the growth, but simultaneously you can see that electricity is also generated. This is a array of the microbial fuel cell, and you can see the lit up LED bulb. Yeah, and the potential being generated, the cell fracturing and healing properties. Yeah, all this work and many more we are working. This is a green future which we are planning, where one room we are having the datum tank, and via our upscaling of the work at IIT Bombay, we have some pillars which are resonating, and they are putting a pressure on the diatoms, and the oil is getting oozed, and via the PDMS layering, the oil is getting absorbed and it's being collected at the bottom. So some hydrophobic sheets and some thermal insulations to maintain the temperature, and the transparent glass for the sunlight to enter. So yeah, so that is the green future. So definitely electric vehicles are not our future, because they will be dumping more of the lithium reserves, and so it is best to be a hybridized electric vehicle. So biofuel and the electricity which is generated from the microbial fuel cells, it can be one way of being zero carbon zero carbon atmosphere. Because even if this fails, the problem is when if the electric vehicles they fail, we have to switch on back to the carbon cool, so which will be a disaster. So that was in the Paris Agreement, which was decided to switch on to the electric vehicles. Well, electric vehicles, I don't know where it will go, but it has a slow start. And if it somehow the wind energy or the solar energy, all this fails, we have to, and if we all have the electric vehicle, we have to switch back to the coal energy, and that's going to be a disaster. So it's better we need to find some zero carbon footprint mechanisms, or maybe diatoms, via which we can have the diafuel run electric vehicles, or the hybrid electric vehicles. That would be better, I guess. So yeah, not only this, we are going to space also, Moon and Mars mission. So and we are working on that also. We are growing datum in the Martian environments. So that's a long way for us to go, where to reduce the payload of the space vehicles. We carry our own food, we manufacture our own food, because green alga, whether it's a hematococcus or the datum, they can be their whole food. And using the pre-treated human waste, or the urine, or the excreta, we can we can cultivate the diatoms or the other potential microalga, which can be, and it can be a great way of terraforming Mars into the planet like Earth, Mars into planet like Earth. So that is the terraformation. So yeah, that's all I think. And these are some the newspaper cutting for the latest patent which we got for the nano fingerprint powder from the Diatoms. So there was gathered a lot of media attention, and definitely this all work is not carried by me alone. This is carried by a bunch of students, dedicated students. This is Ankesh, one of my student who is now in France. Vanna Sroti also, she is in the collaboration work in France. This is Priyanka Khandelwal. This is Gurpreet. This is Urvashi Soni, and a bunch of MSc students are there who are working with me. So yeah, and please join the international conference, which is on September 12th and 13th here in the Hari Singh Gaur International Hari Singh Gaur Center University. This is the international conference, so I'll be happy if you join this conference. Thank you so much.
Sure. Thank you, Dr. Vandana, for this fantastic session and also fantastic PPT. Yeah, maybe you can share us the international conference link or something like that. We can
Yeah, yeah,
definitely
yeah,
forward
yeah.
it to
I'll share.
our
Yeah,
participants. Yeah, fantastic. I think the presentation was so good. So there were a few questions. I think one or two. I would only like to address very few. So one of the question that we got was that can you? I think you already answered it, but can you map the diagrams in a case that for the forensic study especially? I
yeah.
think you already answered this
I already
question.
answered this, yeah.
Yeah, yeah. And I had one question. So talking about on the evolutionary perspective, you were talking about the PCR markers and the biomarkers development. So I think they would also have evolutionary importance, and actually you can.
Climate
Yeah.
change, yes, they do have. They do.
And do you get some like really old diatoms? Like I'm not a diatom expert, neither the plant expert, but uh, like do you get really old diatoms like from, let's say, 200 or 300 years ago or
Uh,
something like
I am not actually a taxonomist, but yes, people are doing work where they are finding as I told you, the Omaru diatoms are there. So
that?
uh, once you find a fossilized diatom, doing their DNA profile won't be possible because the nucleus is not there. But yes,
Yeah.
our taxonomy, you can identify that these belong to which era, or that is
Mhm,
why it's very important in climate change. Diatoms, they also
mhm,
very important in climate change. They
mhm,
help you to diagnose what was the climate during those times. The study is
mhm,
going on in Antarctica mission also. Some people are doing that. I remember.
mhm, mhm. And there is one more question that, like, is there any role of diatoms as substitution of plant-based products? So,
Yes, that's what I told you. The pigments from the diatoms, have so much potential, a higher potential compared to the plant pigments. Regarding the fucoxanthin, whether you take another hematococcus, I told you, not only the
mhm,
hematococcus. When I'm saying that it is having 6,000 times more potential than the other vitamins and the pigments, so it's
mhm,
clear that they are highly potent microorganisms, and their cultivation is occupies less of space compared to the.
yeah, absolutely. So I think we have almost towards the end of the today's session. Thank you so much, Dr. Vandana, for taking time out after the hectic conference
Yeah, it's going on.
things going on, and I'm really looking forward to a second talk or something like that where we can talk about diatoms more, and especially focused on diatoms. I think people are really interested, and me myself as well.
Yes.
So
If I always say to my students, you want to do work on that, and you just you find your field of interest, whether it's the drug delivery, whether it's anything, and you just add that, and you'll get its application.
that's true. That's true. Absolutely. So I would like to request Soumya Mishra to give the vote of thanks today.
Thank you,
Nice from
Dhirya,
you.
for giving me such a nice opportunity. And thank you, ma'am, for providing us such a great, insightful information. Ma'am, I was aware about for the microalgae that they are used for biofuels, nutraceuticals, and in MFC. But this kind of approach of using forensic sciences with microalgae, it's something real like a breakthrough for me, and I would be, yeah, it would be breakthrough for our audience also. So, I'm really thankful from the whole the SciComs Synapse community to provide your time here in your busy schedule and give us a new direction of thinking. And so finally, I would like to thank all of the audience to join us for this such an insightful session for on the diatoms and forensic sciences. So you can visit our website, which is popping on your window, the www.psychomsynapse.in, and to know more about us and to know more about our future events as well. And so with this, I would like to thank everyone and thus signing off. And everyone should have a happy and cheerful day. Till then, please think, share, and grow. And don't forget to subscribe our YouTube channel and please press the bell icon. Thank you, everyone.
Yes, absolutely. It really helps, it also motivates us.
Yeah,
It,
that's
it also motivates us to be in this journey because we also face quite a lot of troubles. Absolutely. Okay. Thank you, Dr. Vandana,
thank you.
again, and all the participants.
Thank you.
Bye bye.